Hey, what's up, AP2 students? It is time for our lecture video on the blood. Now, we're going to be discussing blood today. Blood is part of the cardiovascular system, and the cardiovascular system basically consists of a pump, which, of course, the pump for the cardiovascular system is what? It is the heart, which we see right there. And we got the pipes, which of course are the blood vessels. So I'm talking the arteries, the veins, the capillaries. And we see just a tremendous amount of blood vessels everywhere in this person. And lastly, the topic of interest today is the fluid in that system, which is the blood. So that is what we're going to be talking about, folks. So buckle up. Let's get ready. Let's go. Let's talk about blood. Blood, in terms of what kind of tissue it is a connective tissue. That might strike you as a little bit weird because you might think, well, like bones are connective tissue, tendons, ligaments, there's dense irregular connective tissue. But blood, too, it is a liquid connective tissue. And blood consists of formed elements, which are basically the cells. So, like, this is a white blood cell right here. This is a red blood cell right here. These are platelets over here. Those are all blood cells, also known as formed elements, kind of like an old-fashioned term. And to be honest, we put the word cells in quote marks because, as we're going to see, not all of these formed elements are truly cells. And these formed elements are suspended in a liquid. So the liquid part of blood, which is known as plasma. All right, so blood, fluid connective tissue, cells, which are known as formed elements, suspended in a liquid, and the liquid is called plasma. Now, there are three major types of formed elements. There are the red blood cells, also known as the erythrocytes. This is a red blood cell right here. Notice this kind of donut-shaped appearance where it doesn't actually have a hole in the middle. It just looks like it does. So that is an erythrocyte or a red blood cell. There are also leukocytes. I have a pair of leukocytes in this picture. I got many red blood cells, just a pair of leukocytes. Leuko means white, so leukocytes are white blood cells. Of course, as you probably guessed, erythro means red. Lastly, we got platelets. Got some platelets up here. And yeah, those are my three major types. Notice I've got mostly red blood cells in this picture, followed by platelets, followed by white blood cells. And in fact, that is the order of abundance. In your blood, you're mostly going to have red blood cells. Second most abundant are the platelets. Third most abundant in the blood are the white blood cells. All right. Now, as far as blood goes, it can have two colors. It can be bright red if it's oxygenated. It can be more of a dark red, like more of a maroonish, if it's deoxygenated. So bright red, like scarlet, versus dark red. And it depends on the level of oxygen. Now, you may be like, wait a minute. Let me backtrack for a second here. When I look at this guy, I'm going to notice I've got red and blue as my two colors for blood. Well, that is just to make it easy for you. Like in pictures and models, the red indicates oxygenated blood, the blue indicates deoxygenated. Because just imagine how hard it would be if it were like bright red and dark red. It would be very visually challenging. All right, so that is the color of blood. Another important physical characteristic is viscosity. And blood is generally considered to have 5 times the viscosity of water. So it's 5 times thicker than water. 5 times thicker than water. And that is because of those formed elements, which totally makes sense. Now, let's see. The volume of blood is typically 4 liters in females, 5 liters in males, but there is variation in that. Now, the temperature of blood is usually just over 100 degrees Fahrenheit. So think about it as being slightly warmer than normal body temp. And that is because one of the things that blood is carrying, and transport is a huge job for blood, one of the things that blood is carrying is heat. All right, so we have some physical characteristics. A chemical characteristic is blood's pH range. So a typical pH for blood, like a normal pH range, is 7.35 to 7.45. Remember that pH is the log of 1 over the hydrogen concentration. So pH is a way to measure hydrogens. And because of this equation, I'm not going to have you do any math with this equation, don't worry about it. But notice as hydrogen levels go up, pH levels go down. So lower pH means more hydrogen. Higher pH means less hydrogen. So inverse relationship there. Now, if you have a liquid with a pH of 7, like think about pure water, that is considered neutral. If the pH is greater than 7, then it is said to be alkaline or basic. So blood is slightly basic. Then lower than 7 is going to be acidic. Now, we don't want blood pH to change. If blood pH changes, that can cause proteins, like this protein right here, which has an important fold to it, like a structure to it. pH change can cause the protein to unfold. The fancier word for that unfolding is denaturation, and that's bad. If a protein loses its structure, it loses its function, and that can have serious negative consequences. All right, so blood has a characteristic pH range that we got to know. All right, big functions of blood. Transport. What do we transport? We transport nutrients. We transport wastes. We transport gases. I'm not going to write too many because, I'm just not a good mouse writer here. I really should get like a touchscreen tablet pen thing, but too lazy. And more. We transport more. We regulate. We regulate things like pH, and not just in our blood, but in our body, temperature, volumes, concentrations of things. Think about like electrolytes, sodium, potassium, calcium, that sort of stuff. Blood is also protecting us, primarily protecting us against disease-causing organisms, which are called pathogens. All right, so those are some major blood functions. Let's now just do a little experiment here. Let's suppose we took some blood. We got a volunteer? We took some blood from their median cubital vein right there. Put that blood into a centrifuge tube. Put the centrifuge tube into a centrifuge. Spun it for about 10 minutes. And when you centrifuge blood, it ends up separating based on density. And we can see the 3 major parts of blood. One of them, which we certainly mentioned already, is the plasma. That is the liquid. So in centrifuge blood, the plasma goes towards the top, and it's this kind of like straw-colored liquid. Beneath it, we have a small region, sometimes it's almost too small to see, called the buffy coat, and that is going to contain the white blood cells, so the leukocytes, and the platelets. Buffy refers to like kind of the color. It's like a buff, an off-white color. Then finally, look how big this is in terms of the cells, especially the red blood cells, the erythrocytes. They are going to form this bottom red region. So when we centrifuge blood, we can see these 3 major regions, including the plasma, the buffy coat, and the red blood cells. And the buffy coat consists of the white blood cells and the platelets. Now, we could actually compare the centrifuge blood of a normal person with a person who has anemia. Now, notice that in anemia, in not all types, but in this particular example here, the red blood cell levels have gone down. This is actually going to impair our ability to carry oxygen. Now, if the red blood cell levels have gone up, that is known as polycythemia. Poly means many, cyte is cell, hemia means blood. And this will actually affect the viscosity of blood. What do you think it would do to the viscosity of blood if there were more red blood cells? Would that make blood thicker or thinner? If you said thicker, you're right. It would make blood thicker, more viscous, and that is going to make it harder for blood to flow. So neither of these two situations here are desirable. Now, another thing we got to consider is, what about all these formed elements? Let's get into detail with them. Can you tell me what we got here? What formed element do we have right here, indicated by this red arrow? That's appropriate, right? That is a red blood cell. Over here at the black arrow, what do we got? A whole clump together of platelets. And lastly, I've got, four different white blood cells. This is actually, these two guys over here are one type of white blood cell, another type, and a third type right here. So I got 3 different types of white blood cells here. And let's talk about plasma. Then we'll knock out those white blood cells. I'm anxious to get to the white blood cells. Maybe you can tell. But let's do the plasma. Plasma is the liquid portion of blood. It is 90% water. This is great because water is known as a universal solvent. So think back to your chemistry class. If you're a solvent, that means something is dissolving into you. A universal solvent means you're really good at having stuff dissolve into you. And what kind of things are dissolved in plasma? Well, there are proteins, nutrients, wastes, gases, hormones, electrolytes, and a lot more. All right, we just said that plasma has proteins dissolved in it. Now, plasma proteins have a lot of functions. They help carry stuff. So they do transport. They do fluid retention. What I mean by that is we want the liquid part of blood, we want the water part of blood to generally stay in the blood. Like, we don't want to lose water to our tissue spaces. Think about if someone has really swollen ankles or swollen fingers. That's often because they've lost water from their blood into their tissue spaces in those regions. Now, blood is also going to function in clotting, which is going to help make a patch for a broken blood vessel so we don't lose fluid. And of course, blood proteins are going to play a role in immunity and protecting us from pathogens. And blood proteins also act as enzymes, so they're catalyzing chemical reactions. Now, there are a lot of different blood proteins, a lot of different plasma proteins. We are going to hit 3 major types, including albumin, a group known as the globulins, and then one known as fibrinogen. Now, fibrinogen is a clotting protein. So fibrinogen is a clotting protein. We're going to save this guy for later. He is a clotting protein. We're going to talk about globulins and albumin right now. Okay. Albumin is our most abundant plasma protein. Albumin is going to be made by these cells called hepatocytes, which are in this organ right here. Does anybody know? Do you know what hepato means? So can you recognize this organ? Well, this organ is the liver. Hepato means liver. So hepatocytes are liver cells. So someone has hepatitis, that's an inflammation of the liver. Now, one of the things albumin is going to do, it is going to maintain osmotic pressure in your plasma. Think about osmotic pressure as being a pressure that holds on to water. And so we kind of just mentioned this. We want to make sure we keep enough water in the bloodstream. And albumin does that for us. More on that in a sec. Albumin also does transport. Is going to help transport steroids. So think about like hormones in this case. And speaking of hormones, also helps transport thyroid hormones. And it transports this compound called bilirubin. Bilirubin is a red blood cell waste product. If you've ever seen a jaundiced baby, because they've got too much bilirubin in them. We're going to talk about bilirubin when we talk about red blood cell destruction, but for now, just suffice it to say, albumin helps carry bilirubin as well as steroids and thyroid hormones. All right. So let's talk about albumin's role in osmotic pressure. So I have a blood vessel down here. This is a capillary. And hydrostatic pressure, like the pressure of the water in the blood, which is basically blood pressure, is going to push water out of the bloodstream. So blood pressure is always pushing water out of the bloodstream. I just made a pushing gesture with my hand, which you didn't see. Now, osmotic pressure, which is primarily due to plasma proteins like albumin, is always sucking water back into the blood vessel, especially our capillaries. So blood pressure pushes water out of capillaries. Hydrostatic pressure pulls water back in. Osmotic pressure pulls water back in. So blood pressure, hydrostatic pressure pushes water out of blood vessels. Osmotic pressure pulls water back in, and that osmotic pressure is due to plasma proteins and primarily albumin, just because it is the most abundant. If someone has high blood pressure, they're going to do more filtration. They're going to lose more water. So this can lead to swollen fingers, swollen feet. If someone has not enough albumin, they can also get swollen feet because water's being pushed out, but then it's not being pulled back in. Okay. First, complicated thing we did today. Hopefully that's okay. You're doing okay with that. Let's talk about an example called kwashiorkor. So these poor babies are starving. If you're starving, your level of blood protein is going to drop. If your blood protein levels are dropping, just because you don't physically have as much protein in your food, you can't make it. So albumin levels have dropped. So albumin levels drop. That means the blood has less osmotic pressure. So blood pressure is still pushing water out. So there's still fluid loss going on. But now, because there's not that osmotic pressure, there's not that albumin, it is hard to pull that water back in. And that means there's going to be accumulation of water in the tissue spaces. And in this case, in these children, it is going to primarily accumulate in what cavity? It is in that abdominal cavity. Okay? So this is an example of a situation where we don't have enough osmotic pressure because of a lack of albumin, and there is fluid loss and fluid buildup. And we see it in the abdomen here. There's other things that can do this. People who are chronic alcoholics can get something called ascites. Google that if you want to see that. It is quite an arresting image. Okay. On that note, let's keep rocking right along. We've also got proteins called globulins. And two important types of globulins that we're going to discuss together are the alpha, that's an alpha right there in the Greek alphabet, and the beta globulins. Globulins are generally roundish in shape. They're globular. They're globe-shaped. They are also made by hepatocytes. So albumin was made by hepatocytes. Alpha and beta globulins are made by hepatocytes. Do you remember what hepatocytes refers to? That is cells of what organ? It is this guy right here. It is the liver. The liver. Now, alpha and beta globulins are important in transporting metal ions. So metal, like these two right here. Any guesses as to what two metals I've got right here? Well, this guy is copper. Cu is your abbreviation for copper. This guy is, if we use our periodic table of the elements, Pb, which is lead. So copper and lead. Why did I say lead? I am making mistakes. I'm 20 minutes into this lecture, so I'm not can't redo it. All right, copper was right, and then I misspoke. I didn't mean to say lead. We do not want lead in our blood. I should have said Fe. Anybody know what Fe refers to? Fe refers to iron. Pardon that mistake. It is not the last slip of the tongue I'm going to make during these video lectures. There will be more, always, right? Anyhow, another thing we're carrying is lipids. Remember, lipids are hydrophobic. They don't dissolve in water. We're also going to carry fat-soluble vitamins. So like vitamin A, vitamin K, vitamin E, vitamin D. I always think about the word naked, then take off the N, and you got the fat-soluble vitamins. So vitamins that you would get in this looks like a delicious meal right here. Got some salmon, avocado, a variety of nuts and seeds, and that's like some oysters back there. Fantastic. All right, let's keep on going. So that's our alpha and beta globulins. We've also got these guys called gamma globulins. That's the Greek letter gamma right there. Gamma globulins are also known as antibodies. And what gamma globulins are, they are immune proteins. Gamma globulins or antibodies are immune proteins. They are made by white blood cells called lymphocytes. This is a lymphocyte right here, and they're made when you're having an immune response. And what antibodies do is, here's a bacterial cell, a pathogen. These green guys are antibodies. They grab onto it and they mark it for destruction. They tag it so that other cells can come along and say, oh, you're tagged with an antibody. I'm going to eat you, or I'm going to make you explode. All sorts of cool ways we do that. We'll come back to antibodies when we do the immune system. All right, so those are gamma globulins. We did alpha and beta globulins. We did albumin. We are doing great. Now, another component of the plasma are these things called buffers. Now, buffers are chemicals that resist changes in pH. Do you remember what the range for plasma pH was? 7.35 to 7.45. And that's a really important range that we want to maintain. Buffers help us maintain that plasma pH. And we know if pH changes too much, proteins can do what? They can unfold. And our other word for unfolding is denaturation. Now, two of the major buffers we have to be aware of are albumin, as well as this chemical compound called bicarbonate, HCO3-. These are two really important buffers. So albumin is doing a lot of jobs, right? It was maintaining plasma osmotic pressure to hold on to water. It was carrying steroids, carrying thyroid hormones, carrying bilirubin. All sorts of important jobs for albumin. It's also acting as a buffer here. Fantastic. Quick little thought problem here involving buffers. Suppose I had two solutions, solution A, solution B. I added 10 mils of HCl, hydrochloric acid, strong acid, to each of these solutions. After I added to the acid to the solution, right, I'm adding acid, and so what do I expect pH to do? I expect it to go down. And solution A's pH dropped by 4 units. Solution B's pH dropped by 1 unit. So which solution was the better buffer? Think about it for a second. Go ahead, pause the video if you want to think about it for a moment. And here's the answer. Buffers are going to prevent pH change, right? They're going to reduce pH change. So you got to ask yourself, which solution had the least amount of change? And the least amount of change would be solution B. So solution B would be the better buffer. All right, good job, good job. Hopefully you got that right. If not, that is cool. Make sure you understand it. Ask me questions if you don't. And with that, folks, we are done with plasma and the plasma proteins. So now we can move on to our formed elements, starting with our red blood cells, our erythrocytes. And let's see if we can talk about erythrocytes, then we'll call it quits for this first lecture on blood. So erythrocytes are also known as red blood cells. You typically have about 5 million per microliter of blood. And by the way, a microliter of blood is a millionth of a liter. So think about the math here. Like, you've got 5 million red blood cells, right? Per microliter. 5 million is 5 times 10 to the 6th. All right? And you've got how many liters of blood? Well, you probably have 5 liters of blood, right? So 5 liters of blood is 5 million microliters of blood. So we got 5 million. We've got 25 times 10 to the 12th red blood cells. That's how much you might have, 10 to the 12th. That's a huge number. That's crazy, right? All right, that's all the math for right now, I think. I know you love math, right? I know you do. Don't worry, I like math too. Okay, so the shape of a red blood cell is known as a biconcave disc, and concave means punched in, bi means twice. So we look at these red blood cells in this electron microscope, and we can see that they are punched in twice, right? They're punched in twice. This biconcavity makes them really flexible, which is good for twisting and squeezing through narrow capillaries. It also makes them stackable, so they can kind of like almost line up in a row, like go single file through narrow capillaries. Now, red blood cells are also said to be anucleate. So the prefix A means without, nucleate means nucleus, so they're without a nucleus. They also lack organelles. So there's no like endoplasmic reticulum, no Golgi apparatus, no mitochondria. And this is actually really important, as we shall see. Okay, so red blood cells last roughly 4 months before they die. All right? They can't divide to make new ones because they're anucleate. They lack a nucleus. They can't like repair themselves because they lack a nucleus and they lack organelles. So there's the downside of, this lack of nucleus and organelles. An upside is there is a lot of space in these guys. And that space can be filled with a protein called hemoglobin. Hemoglobin is our oxygen binding protein. Hemoglobin can grab onto, can hold onto, and then can let go of oxygen. So red blood cells, because of their hemoglobin, red blood cells are the oxygen transport cells. They transport 98.5% of your blood's oxygen, as well as a significant amount of your blood's CO2. So hugely important jobs, especially the oxygen transport for our red blood cells, our erythrocytes. Now, what we see right here is an example of that stackability. So because red blood cells are biconcave discs, they're stackable. So they end up getting single file in our capillary here. And this single file like stack of red blood cells gets a French word. It's called a rouleau. So it's a rouleau of red blood cells. Okay? Pretty cool. I like that picture. It's just neat looking to me. Okay. Now we hinted at this a little bit ago, but red blood cells are a tremendously abundant cell. 25 times 10 to the 12th. Yes, I did my math right earlier. It's 25 trillion. You might have 30 trillion cells in all. And oh my God, 84% of your blood cell, of your not your blood cells, of your cells are red blood cells. And platelets are your next best here. Bone marrow cells, blood vessel lining cells, other cells. You know, like look how many muscle cells you got. It's not that many. You know, this is crazy how many red blood cells you have. Like what, you know, how much of you, me too, we're the same here, made up of red blood cells. All right, if I look at these red blood cells here, what are these red blood cells missing? They are missing, what are they missing? They are missing a nucleus. What else are they missing? They are missing organelles. That says organelles. That's me writing in cursive. And what's your favorite organelle? Probably mitochondria. Now the downside of lacking the nucleus and organelles is you can't, make new proteins, you can't divide. The upside, we mentioned an upside earlier. An upside was that we can have more space left over to carry oxygen because we can fill the cells with more hemoglobin. But there's another upside. Red blood cells lack mitochondria. Red blood cells lack mitochondria. All right, why am I breaking that up? Okay, do you know what happens in mitochondria? We make ATP, which is a source of energy, right? We use nutrients and we make ATP. Do you know what else gets used up in mitochondria? Oxygen gets used up in mitochondria. So if red blood cells have no mitochondria, they cannot use oxygen. Analogy I like to use. It is, I don't know what time you're watching this video, but as I'm making it, it is like right around 12 noon, lunchtime. I am definitely getting a little hungry. If I were hungry, would you want to give me your pizza? And say, hey, would you mind carrying this pizza? Like not to like eat, but would you mind having me deliver your pizza? Be like, hey, can you take this pizza upstairs on your way up? Drop it off at room 212? You would absolutely not do that because what would I do along the way? I would eat the pizza, right? You never let a hungry person carry your food. It's the same idea with these red blood cells. If they had mitochondria, they would eat, I put that in air quotes, they would use up the oxygen. By not having mitochondria, they don't use the oxygen. They're better at transporting it. That is really cool. Okay. What else do you notice about these guys? It's something we mentioned earlier. A lot of red blood cells look like they're empty in the middle, right? Like they're a donut, and they're not actually empty in the middle. It's just that biconcave shape makes them so skinny in the middle that light abundantly goes through and they appear empty under the microscope. All right, good to know, good to know. Let's keep going. So we know red blood cells are stuffed with hemoglobin because of the lack of a nucleus and organelles. Hemoglobin is this protein. Whenever you see globin, like kind of almost reminds you of globulin, right? Think protein. And hemoglobin literally means, really refers to blood and round protein. The hemoglobin protein is made of 2 alpha chains and 2 beta chains, which are the chains are made of amino acids. So like this guy right here, this sort of darker brown ribbon is a beta chain. Another one over here. I got an alpha chain down here, another alpha chain over here. Each of these chains has this chemical structure called a heme group. This red disc is known as a heme group. So I got 4 chains, and therefore I got 4 heme groups. This is a heme group over here. You don't have to memorize this chemical structure, don't worry. But you do got to know that in the center of each heme group is an iron. And this iron, which is represented by this little like bluish circle here, this iron is really important because an iron can grab onto a molecule of oxygen. So every hemoglobin protein has 4 hemes, so it has 4 irons, so it can carry 4 molecules of oxygen. Now you might have a 250 billion, 250 million hemoglobins in a red blood cell. So if you have 250 million hemoglobins in a red blood cell, well, times that by 4, you could carry 1 billion oxygen molecules. Crazy numbers we're dealing with here. Okay, now if hemoglobin has oxygen attached to it, we refer to it as oxyhemoglobin. Okay? If hemoglobin does not have oxygen attached to it, we refer to it as deoxyhemoglobin. Now, we also have a word for hemoglobin when it's carrying CO2. That's called carbaminohemoglobin. Now, the CO2 and the oxygen, they're not carried in the same place. So you can be carbaminohemoglobin and you could be one of these guys. All right? You can't be both oxy and deoxy, but you can be either of these guys and you can be carbaminohemoglobin. So carbamino refers to the fact that it's the chains where you carry the carbon dioxide. And remember, it's the iron where you carry the oxygen. I do want to point out that carbon monoxide, which is a poisonous gas, binds really tightly to this iron. So this is why you don't want to, be in your garage with your car running, because the exhaust makes carbon monoxide. You don't smell it, you don't taste it, but you breathe it in, it goes into your blood, it binds this iron, and then oxygen cannot bind there, and that can kill you, that can be fatal. All right, fun little tidbit of information there. Sorry to bring it down. Okay, I was hoping to finish these red blood cells. Let's see, how many more slides do we have on red blood cells? We kind of got a lot. You know what? I might call it a day on this first video lecture. We're almost at 40 minutes. I don't want to go too long for both you and for me. So next time we're going to pick up with hemopoiesis, which is blood cell production. So I will see you guys next time. If you made it all the way to the end of the video, I always say this because people don't watch the whole things. Let me know you get to the end. Tell me you got to the end. I will be impressed. I will applaud you for that. But yeah, I'll see you next time. We're probably going to do 3 videos on blood overall. First one is done. See you later.